using 3 pigs being fed levels from 50 to 2,500 ppm of zinc oxide or zinc from zinc oxide in the diet. And you can see you can cluster all these 3 piglets depending on the dietary zinc level. So there must be a very strong impact of this trace element on the composition, on the diversity of the gut microbes. And we have done that also by other methods using 16S RNA sequencing. And what I found quite interesting was, without going into any detail, that you also have a clear impact of these different zinc levels, for instance, on smaller bacteria groups. So we are always talking about the core microbiome, but we always neglect all these smaller groups, and obviously they are very much affected by these different dietary zinc levels. The question was, what is really relevant? For that, we made a differentiation. So looking at the total zinc in the GI tract, the insoluble part, which was bound to the matrix, the so-called free part of the zinc fraction. So we extracted, this was extractable from the matrix, and we also looked for the effect of protein-bound zinc. And just to show you, although we have very high levels of total zinc in the total GI tract from the stomach to the colon, we see that the soluble part is much lower, but it seems to be somehow higher in the stomach, which can be explained by the pH. But really interesting is, from my point of view, that when we consider the correlation between these free zinc levels with the GI microbiota, you see there are some reasonably high correlations which were not detected for the total zinc and not so much detected for the protein-bound zinc. That means finally that very obviously the free part of these zinc ions is really responsible for the effects on the microbes. For me, there's a very interesting observation. Maybe I can explain it briefly with this slide. So again, we have low, medium and high levels of zinc in the diet. And for instance, when you see here the concentrations of lactobacilli in the GI tract of piglets, you see at low levels you have the highest levels of lactobacilli. It goes dramatically down when you increase the dietary zinc level. On the other hand, we see more Clostridia. We also see more gram-negative bacteria, E. coli, Shigella group. So that means finally it's a paradigm somehow we get with high zinc levels, somehow less problems with gut disturbances, with diarrhea, but we really decrease the so-called beneficial bacteria and increase somehow the enterobacteriaceae. So we make them more diverse also, which is quite relevant. So the next question we were asking is, has this any impact on other functions in the gut, especially on the occurrence of resistant bacteria? And we know from the literature that zinc resistance is somehow related to antibiotic resistance, which means that there could be a link, and actually this has been shown by one of the doctoral students here in the Institute of Microbiology in our university. So what she described in these piglets which were fed a high level of zinc oxide was there was an increase of E. coli genetic diversity, and there was also a significant increase of multi-resistant E. coli compared to the control group. So we had about 19% of multi-resistant E. coli strains in this high zinc oxide group compared to 0 in the normal group. We saw the same for tetracycline and sulfonamide resistance. It looks very complicated, so but in general there was a statistically significant increase. And just from some recent studies that we did together also with some companies, we've seen here directly after weaning of the pigs being fed low or high levels of dietary zinc, that we get some differences in the levels of E. coli after weaning, which is quite higher here in the group getting these lower levels of zinc oxide. But very interestingly, we saw that there is a very rapid reaction of these piglets after weaning when we consider here the tetracycline resistance gene concentration, which is high directly after weaning, goes down and then goes up again We see higher levels in these high zinc oxide piglets. We see the same for sulfonamide resistance genes, and we see the same also for quinolone resistant genes. So that means finally, completely unexpected for us, we see that we have an impact on bacterial diversity, but we have also an impact on these factors which may be relevant not just for animal health but also for consumer safety. So, considering health, there's in pigs no doubt about the short-term protective health effects in piglets, which is documented. We have clearly a legal situation which prohibits the usage of high levels of zinc oxide in animal production, but Denmark and Belgium have some special regulations nowadays. In both countries you can use zinc oxide in high levels after prescription. In other countries it's possible, but requires some, let me say, fantasy. So we have also in Europe a high number of unauthorized uses of these feed additives going up to 5,000 ppm, which means the risk for antibiotic resistance development may be even more than we see it from our data. And very clearly we have to say one point is that we have an impact on these resistant genes. We have an accumulation of zinc in the soil in different areas of Europe, so which may have a negative impact on plants in the long term. And the final conclusion is, does zinc have an impact for the industry? What we experience is there's a high interest, really worldwide interest in this topic. What we can say is, what we were able to confirm, and others have done it in much more depth, that it is essential, no doubt about it. We have also a clear indication that it has protective effects on animal health, but we need other solutions, and I think this is something we have to consider also in these projects that we are discussing. So I think the mechanisms that we were able to identify how zinc could protect the animal from getting ill, from developing diarrhea, are highly relevant and highly interesting, but I think clearly we need to go in a different direction, because the ecological aspects and this antibiotic resistance topic is, from my point of view, not good enough to weigh out the protective effects. So this is maybe stuff for the next project. Thank you very much.